So welcome everybody. We will start the data management workshop for the 2022 and 2023 Biodiversa funded projects. Welcome from Biodiversa side. I will welcome here Rainer Sodtke, Biodiversa co-chair, who will give you first the welcome words.
Thank you, Harri. Yes, a very warm welcome to this workshop from Biodiversa Plus European Biodiversity Partnership this afternoon. I think it's the second data management workshop we are organizing within the Biodiversa Plus partnership now for the second call, the Biodiv Mon 22-23 joint call projects. And so a very warm welcome to everyone attending this afternoon. I see we are around 60 participants at the moment. I think some 80 or 90 registered. So I hope that there will be also some colleagues joining in a few minutes. I'm also happy to announce that we'll have a few speakers this afternoon. We also have Gaby Rerig from German Research Foundation, DFG, with us, who'll present in a few minutes, and Jennifer Anderson from the Farm Action Project, and also Dimitri Brosens from IMBO, which will present later. And later in the afternoon, there will also be presentations from Birgit Gemeinholzer from GBIF, Melissa Liu, also from GBIF. And I think that's it for a short overview. Next slide, please. Okay, this data management workshop today has, of course, different objectives. As I said, the project participating in this workshop are the projects of the second Biodiversa Plus call. We launched in 2022 and 2023. And the projects started earlier this year, and we had just a very successful kickoff meeting in Tallinn in April this year, where all the 33 projects were able to present, and it was really amazing to see the huge array of topics you're covering with these projects. And yes, today we want to focus on data management within all the projects, want to exchange on best practices on this issue, come up with recommendations on the data management plan, which of course should be in line with our Biodiversa Plus internal data policies. So we have specific objectives to cover for today. For your information, of course, we want to make sure that all the projects understand very well the principles and the concepts around open data policies around data management and data availability in the projects. So in order to all use the advantages of having a well-structured and complete data management plan and data policy within the projects. And of course, we also want you to give support and maybe also to build up capacities if needed, in order to provide you with all necessary information and advice you need in the projects to manage your data, to ensure open data availability, any important information you need to set up data management plans within your project. And of course, we're happy to answer every question you have around this topic you might identify around data. And so, yes, to inform you on all aspects here and on the relevant people who can give you this support actually. For the Biodiv Mon projects, please let me remind you that you are asked to send up your updated data management plan after the workshop. We have set a deadline, which is the 21st of July, so a bit of time ahead on this. And please send this to Harri Hautala, which already welcomed you a few seconds before. I'll send this to Rerig, please. Okay, I think next slide, Harri, it's your turn again.
Okay, thanks, Rainer, very much for your very nice introduction and good words. My warmest welcome also to the DMP workshop. I'm Harri Hautala. I work as a science advisor at Research Council of Finland in Finland, as you can guess, and I have been building up this workshop together with my colleagues from France, Germany, Italy, Poland, and many, many other countries. We are very happy to see you so many here. We have almost 100 persons registered to this little event. This is actually almost double as much as last year when we had the first actual workshop of the Biodiversity European Partnership. Thanks for sharing your time. We have both funded researchers, but we have also data managers from the second Biodiversity Partnership joint call. You have also shared with us in the initial registration some of your current knowledge level on data management, and here are the results. Next slide. Yeah, the sliders here. As you can see, we have experts. We have 11 experts, 11 who have confessed that they are experts. We have a lot of people with good experience and moderate experience, but we have also people who are not so experienced with data management issues. For Biodiversa view, we will see that good and well-functioning data management, it is fundamental for the quality of biodiversity research. And we have really, just like Rainer said, we have set up this workshop for the funded researchers from this second call to help you with your DMPs. Because we as a partnership, we strongly support open science, open sharing data, open access publishing, and all those fair principles that you will hear quite soon. Okay, the next slide, please. Thanks. On this slide, you can see the names and affiliations of today's speakers. Rainer also already kindly listed some of the speakers here. So just to repeat, we have Rainer, we have Gaby, me, we have here as almost the most important technical persons in the meeting. We have Céline, we have Sophie from France and Germany. Then we have our most important persons for your knowledge. We have the speakers. We have Jennifer Anderson from Project Fun Action, which was funded by Biodiversity Protocol. We have Dimitri Brosens from INPO Belgium. We have Birgit Gemeinholzer, who is GBIF Science Committee Chair, and we have also from GBIF from Asia support team, we have Melissa Liu. So welcome all. We hope really now here that this will be a very useful event for our research work, and we hope that you both learn and also enjoy, and not just learn, but also enjoy from the presentations and from some breaks and mentis we are going to have. I will list mention here some practical things before we will start. You can present your questions and comments after the presentations. We have allocated some time after this. The whole meaning of the workshop is to really help and advise you with the DMPs, which can be tricky sometimes. You can leave your questions and comments also in the chat. And when you speak, please keep your videos on so that it is sometimes easier to hear when we will see also your face. And we are also then much more interactive and alive here. So, and we will be really recording this event, but afterwards it will be shared only for the participants of this workshop. So if you sneeze in the workshop, you cannot find it from public YouTube afterwards. You have all received the agenda of today. It was sent with the registration. We have 4 parts basically. We have this introduction session, which is soon ending. Then we have presentations session, and we have some mentees in between. We are asking you some questions, and we will take a look at the answers. And we have a small concluding part. And of course, because we are people, we need a biological break in between the program. Finally, I would like to remind here you also that this is a two-part workshop, as you know. Tomorrow, this workshop will be extended with a special capacity building workshop on Darwin course number, and this was organized by our other Biodiversa colleagues. But it will start tomorrow morning, but more about that at the final part. So now it seems that Gaby is there, and to lighten up the start of this workshop, we have some icebreakers from Gaby. Please, stage is yours.
Yeah, thank you, Harri, and welcome from my side. I'm Gaby Rerig from the German Research Foundation, and as said, we would like to give you a light start in this workshop with an icebreaker. So you will soon see 6 pictures, and you can choose one of these pictures, and you should answer with this picture the questions: data management plans, how do I feel about it so far? You can take the question as you like. So you can either refer to the process, how to write a DMP, or you can think about what do you think is the outcome of a DMP. Is it positive? Is it negative? About your general feelings, just as you like, as it's just for fun. So Sophie, please start the Menti for us. Sophie will copy the link in the chat, and then you are all invited to participate. And yeah, when we see the results, I will, of course, I will not know why you have answered or why you have ticked a picture, but I will tell you a bit about my associations. And then I will also go with these pictures through our agenda. And yeah, you will see that hopefully everyone, will benefit from our workshop. So I think we can keep the Menti still open if someone else would like to give his answer. And I start with the first picture. So yeah, I see 6 pictures of 6 people of you think, oh dear, DMP, it's just another heavy burden on my desk. There are also so many tasks on it. So yeah, of course, with this workshop, we would also like to motivate you for your DMP, to do it for this project, but also in the future. And so to give you motivation, we will start the workshop with an enlightening video with a panda. And this video is about data sharing and management. Yeah, so the second option, the picture with the steps, maybe you are more interested in the practical aspects, how to write a DMP. So yeah, we would like to with the first presentation to give you practical insights. So we have, as you have heard, Jennifer Anderson from the Biodiversa Biodiv project call with us, and she will give us a presentation, data management plans, why scientists should create and sustain them. And so let's hear from the steps her consortium took on this excellent. Yeah, the next picture, you feel helpless in the jungle of information and a lot of terms and conceptions. And for this, we have Dimitri Brosens with us, who will give us a first guided tour through the jungle with his presentation, Open Science and Open Data, Main Principles and Concepts. So the fourth picture, 6 people, maybe they are not sure about DMP, how they should feel about it. On the one hand, of course, open data, fair data, that sounds very good. But on the other hand, okay, we have to invest a lot of effort in it. So is it will this data really be used in the past, or is it maybe only a luxury data cemetery which will be built up. And yeah, of course, we would like to give you a positive view. And from this, we have got Birgit Gemeinholzer with us. She is, on the one hand, a colleague from you because she is a coordinator in the Biodiversa project. But on the other hand, she is also chair of GBIF Science Committee. So GBIF, the Global Biodiversity Information Facility, and she will tell you some success stories in her presentation, data management and open data in GBIF. Yeah, I think, the fifth option with 17 people is really liked. You are looking for a toolbox, how to write DMPs. And for this, we have another speaker, Melissa Liu, also from GBIF, and she will present GBIF training package on data mobilization. And this is also, we are planning a course on this later during the lifetime of the project. It will probably be in the first or second quarter of the next year, a virtual course where you are free to join. So here you will get a first taste on it. And yeah, as so many people are interested in a toolbox, I hope you will also join the course later the year. Yeah, and the six options, very positive already. Maybe you think, yes, this open data, fair data is really good for science, for knowledge generation. We'll go through the roof with it. So yeah, we hope that at the end of the workshop that this will be your feeling about DMPs. Yeah, and the magic drink of our workshop, as said, we've got these 4 presentations with time for Q&A after this. We have got interactive part with mentees where you also will have the chance to comment a bit on your answers, not as here where I did it, but where you can tell us a bit about your experience, expectations, about your views on a topic. And yeah, we of course will also have a virtual break, as Harri said, and tomorrow the second part of the workshop. So now I hope, or now you will get another night start in this workshop with this Panda video. It's 5 minutes and it's all from 2012. So let's see whether it's still up to date. Get a relaxed position in your seats, and Harri, I think it's you who will start the video for us.
Yes.
Hello. My name is Dr. Judy Benign. I'm an oncologist at NYU School of Medicine.
Hello, Dr. Judy Benign.
I read your article on B-cell function. I think that I could use the data for my work on pancreatic cancer.
I am not an oncologist.
I know, but I think I could use the data for my work on pancreatic cancer. Do you have the data?
Everything you need to know is in the article.
No. What I need is the data. Will you share your data?
I am not sure that will be possible.
But your work is in PubMed Central and was funded by NIH.
That is true.
And it was published in Science, which requires that you share your data.
I did publish in Science.
Then I am requesting your data. Can I have a copy of your data?
I am not sure where my data is.
But surely you saved your data.
I did. I saved it on a USB drive.
Where is the USB drive?
It is in a box. It is in a box at home. I just moved.
But can I use your data?
There are many boxes. So many boxes. I forgot to label the boxes.
Hello again. Thank you for sending me a copy of your data on a USB drive. I received the envelope yesterday.
You are welcome, but I will need that back when you are finished. That is my only copy.
I did have a question.
What is your question? You might find the answer in my article.
No. I received the data, But when I opened it up, it was in hexadecimal.
Yes, that is right.
I cannot read hexadecimal.
You asked for my data and I gave it to you. I have done what you asked.
But is there a way to read the hexadecimal?
You will need the program that created the hexadecimal file.
Yes, I will. What is the name of the program?
Cytosynth.
I do not know this program.
It was a very good program. The company that made the program went bankrupt in 2007.
Do you have a copy of the program?
I do not use this program anymore because the company that made it went bankrupt. Maybe you can buy a copy on eBay.
I have good news.
You again.
I talked to my colleague. She knew a person with a copy of the software.
Then why do you need me? Everything you need to know about the data is in the article.
I opened the data and I could not understand it.
If you have the program, you will find it is clear.
Well, I noticed that you called your data fields SAM. Is that an abbreviation?
Yes, it is an abbreviation of my co-author's name. His name is Samuel Lee. We call him Sam.
I see. And what is the content of the field called SAM1?
Ah yes, SAM1 is the level of CXCR4 expression.
And what is the content of the field called SAM2?
That is logical if you think about it.
What is the content of the field called SAM2?
I don't remember.
What about SAM3? Is there a guide to the data anywhere?
Yes, of course. It is the article that is published in Science.
The article does not tell me what the field names mean. Is there any record of what these field names mean?
Yes. My co-author knows what the content of SAM2 is, and SAM3. And SAM4.
Can I talk to your co-author?
I'm not sure.
I would very much like to talk to your co-author.
Well, he was a graduate student. He went back to China 2 years ago.
Can I have his contact information?
He is in China. His name is Sam Lee.
I think I cannot use your data.
You could check the article to see if what you need is there.
Please stop talking now.
Okay, it seems that the video was there. Okay, I hope you really enjoyed it. I actually added the link, the YouTube link, to the chat box. It seems that it is still relevant one for some people at least. And you can share it with your own data managers, with your own projects, because it is a nice video that we have already shown for the last workshop, and actually a workshop also before that. So thanks, Gaby. Now we are moving to our presentations, and we have them from 4 wonderful people who promised to help us in this workshop by giving their extensive knowledge on data management issues. Audience can, you researchers and data managers, you can present questions in the chat, or you can raise hands, but preferably after each presentation, because we have saved 10 minutes after each presentation. So we have 20 minutes for the presentations and 10 minutes for questions and answers. A small reminder also for our dear speakers that I will give a yellow note somehow when you have 2 minutes time, so that we can keep the basic time schedule. It seems that we are exactly in schedule at right now. Okay, so as a first speaker of today, we have Jennifer Andersson, who is a consortium coordinator of Project Fun Action from Biodiversa Partnership's first funded call Biodiv project. Title of her talk is Data Management Plans, Why Scientists Should Create and Sustain Them. So, as Jennifer's consortium was funded from our previous call, and after we have checked all those updated DMPs from those consortiums, we noticed that Jennifer's consortium's DMP was an excellent one, and that's why we asked Jennifer to present to give this presentation in this workshop.
All right, thank you for this opportunity to talk to you today about data management and some of the insights that we've been gathering in the first almost year and a half now of our BioDiv Protect project Fun Action. For reference, when we did the Menti last year for this at the DMP, I was in the I was on the left of that range of distribution of scientists and how they felt and then what their experience was in data management. So I've had the opportunity to grow tremendously in the last year, and that's largely thanks also to the Data Management Committee, who are a part of Fun Action, and I'll show you those names at the end. All right, so I want to introduce you to the Fun Action Consortium. So we are 8 partners who are indicated here in bold. We represent 7 countries. We have 6 national funding agencies. We also have 1 in-kind funder, the Global Center for Species Survival at the Indianapolis Zoo. We have 25 current participants, and that's growing by at least 2 in the next couple of weeks. That's just to give you some of the structural information, and that will, I'll fit this together here in just a few slides. So what are we cooperating to achieve? We're working together towards the conservation of aquatic fungi. So I think some of you have possibly already heard a bit about these fungi from Andy Bruder, who's the coordinator of Most Fun, which is a BioDiv Mon-funded project. But for context here, I'll just say a few words. So there are more than 6,000 known species of aquatic fungi. They represent the diversity of the fungal kingdom. And they play very important roles in our aquatic ecosystems as decomposers, predators, pathogens, parasites, and more. But these organisms are currently not part of any major conservation, planning, or action. And so that's what the Fun Action Consortium is overall working to change. And we're working to change that through 3 seemingly simple-sounding approaches by building a foundation of knowledge, by translating that knowledge into conservation actions, and through communicating to build support for aquatic fungi-aware conservation. And this is just the shorthand framework that we can think about, that we can branch out from to see how the project is structured. So these overall 3 objectives are implemented through 5 work packages, and I'm sure there's some kind of similar structure in most of the projects represented here today. Each of our work packages is co-led by pairs of partners. Most often these partners are located in different countries and have their own teams as part of the overall Fun Action project. These work packages are highly synergistic. So we have collaboration and data flows that happen between the work packages, which means this is data flow and collaboration between countries and teams. And so really among all of the 25 people that are currently part of Fun Action. So for this system to work, it's absolutely vital that our data is produced in one work package, can be efficiently, confidently used and implemented in the other processes and analyses in the other work packages. It's also important that we do this in ways that don't introduce errors or flaws or unexpected changes or unknown entities into the data by people who are working in different ways at different times and at different places and at different data cultures across the different universities. So what kind of data are we generating? At the most basic level, or the easiest for me to connect with the level, is the data that we collect when we take samples. So we are taking samples of aquatic fungi from watersheds at 6 countries. So this is actions taken by 6 different teams. And again, probably most of those 25 people. At each time that we sample, we collect interrelated sets of samples. So that means we have to have some knowledge that's trackable throughout the project that allows us to know that this set of samples was taken from which site at which time, in which watershed, and what is the content of that sample. And this information has to be transferable so that we can use it. Let me just grab my highlighter here, so that we can use it going from the samples themselves through the continuation of generation of data about those samples as we generate additional primary scientific data like DNA sequences, the site metadata, water chemistry information, and these kinds of things. And that has to stay trackable as we go then to the additional secondary scientific outputs that are the analysis products of this work. So I have to say, this is kind of the land where I am the most comfortable or had had the most experience prior to this project. But as you can see, we are generating and using a lot of additional types of data. And it's important that our data management includes these considerations as well. So I think many of us, as I said, are most familiar with the data types on the left, probably also the import and use of independent data sets, whether those be public or not yet published. But in Fun Action, for example, we have a lot of data in the form of photos, both with and without people, all of the consortium level documentation that is happening. This includes the personal information about our participants, photos of our participants, their social media account names, their titles, positions, work addresses, and so on, all of which is personal data. We interact with our national funders. We have contact information and logo rules and all kinds of information about that. We are collecting and actively working with our information about and actively working with our stakeholders. So we have a collection of stakeholder information. We use surveys as follow-up tools for our outreach activities. We have webinar registrations. We have all kinds of outputs from these kinds of outreach activities that aren't kind of in the purview or in the mind, I think, of many of us when we're just thinking about more of the data gathering and analysis parts of our work. Just a few special things to highlight in the data that we're collecting is that we are collecting very large data sets. So because of our sequencing activities, we're generating tens of terabytes of data. This requires redundancy and secure storage. And we're also generating more personal data than I think I had really appreciated until we really got into things. And of course, this personal data means that we have to have some understanding and ability to apply and fit to the regulations of the GDPR. And so this requires, for example, not storing this information in cloud-based instruments, having restricted access. So this means things like our Google Drive are not appropriate. And so finding different ways to deal with these kinds of data. So these have been some of the things actually that have been really helped. Dealing with these issues have been really helped by the GDPR planning that we've undertaken. So why have I shared all of this with you? Of course, we're passionate about this, the topic of the work that we're doing, but it's just to give you the context of the complexity of the systems in which we're working and in which you might be working now with your Biodiversa projects. So we have a single project with 8 partners, 7 countries, 6 national funding agencies, 25 team members, 5 work packages, each with 2 leads, which means work being done or led in at least 2 countries. We have 10 case study watersheds, 8 sampling times where we sample those watersheds. So far, we have completed 448 unique site visits for collections, which has resulted in more than 2,000 samples. And all of that has to be consistently collected in a standardized way and an organized way so that we can collaborate best between and among our partners. We have personal data, we have unique raw data that is a very large size. We have documentation, some of which remains internal, but much of which we have to have appropriate ways to share that in the public sphere. We have this highly synergistic system of interrelationships between the work packages and countries and leaders and teams and researchers. And all of these things have to work in a context that fits so that we can achieve our goals in terms of open science. And I'm not going to talk much about the open science aspects because I know we have quite a agenda. That's quite a full topic on the agenda today. So not only is this kind of the framework in which we're working, but we're also sending these samples, the actual physical samples, to different countries and having analyses performed at different organizations. So we have to have our information from the start of our project, from the very moment of collection. We have to have that fully traceable, usable, and shareable for all of the analyses that are taking place in all of the different places. So we, for example, send our water samples to Spain for chemical analyses, where we have, I don't know, 24 or something like this, chemical parameters being estimated. We send all of our physical samples, all the other physical samples to Estonia, where they will be part of processes there. They send those back to Sweden, where they get sent for DNA sequencing. And in the end, we have to be able to put all of these parts together in a way that is uniform and standardized and usable and transferable among all of the actors. We also have, and that's, sorry, what I indicate up here with all these arrows between all the work packages. So some days, if you want to ask me why we should create and sustain data management plans, this is my answer. It is the avoidance of chaos. So there's many opportunities for things to go wrong, for issues to be introduced into data sets and data collections and processes that are really going to make our work in these projects difficult. And so I think any actions that we can take to avoid that are beneficial. However, I often prefer to think about the data management planning in these more positive terms, because to me, these actually are really more fully and more accurately reflecting the experiences that we've been having in the making and using and updating of our data management. And I'm not sure I would have believed that I would be so positive about this today when I was sitting in your shoes last year. But this is really shown to be true in our case, that this, through the processing and through the planning and the production of the data management plan, we have developed a better way to work efficiently together, to have structured data collection and storage, to reduce the risk of data loss, to improve consistency within and among the teams. And this includes over time, both within the project lifetime, where we might have actors who come and go in some of our shorter term roles or employments, but also to allow us to transition the data into those open source platforms. And produce it, and in that way, it also promotes the sharing and reuse of data and reproducibility. We will have continuity. We can track what's happened to our data across time and across groups, and the longevity, so that supporting the deposition into the public databases. I would just note that I have modified this content from the Biodiversa's products that they produced, which I can say have been their data management plan booklet, which I think I've read multiple times and multiple versions multiple times because they have actually been very helpful to me. So thank you for that. So just a few things about the Fun Action Project and our approach to the data management planning specifically. One thing that we have thought about the data management plan and planning, in addition to the things that we just talked about, is this, or are these rather. We see that the data management plan supports common understanding and expectations among the partners and their associated teams. For the management of the data associated with the project. So we establish this common understanding and expectations already from the beginning, so that there's not confusion, there's not competing ideas later. We can improve things, but we start from a standing base. It serves as a tool to ensure that we meet the research community, partner institution, national and EU standards for data access. This includes the FAIR principles, which I'm sure we will hear more about today, and supports our deposition into these appropriate public repositories, again, for this longevity and reuse of the data. And it provides a mutually agreed framework defining how we use and share and store the data. So we know who's responsible for the data storage, where it will be stored, how much it costs, what is the redundancy, what are the backup systems. All of this is agreed. And again, all of these things can be adapted and changed. That's up to us, but we start from one standpoint. And I think that's a very efficient and effective and practical and pragmatic way to work within a consortium of this size. So I'm very proud of the work that we've been doing. You can see our first draft, our first version of our data management plan is on Zenodo. And so you're welcome to check it out there. But this really is a living document, and we already know, for example, what things we want to work on next, and what changes we would maybe like to make to some of the existing content, and we're using it in this very live way. So we have just a few minutes left, and I thought I would share with you a few ways that the data management planning has been helpful to us so far. So one of them I already touched on, which is we have in place, we know it's already established, we know who's paying, where it is, how it's backed up, and so on, these extremely large data sets. So we have the safety and stability of the data storage for these very large, irreplaceable data sets organized from the beginning. And then it's really been helpful in us working through some of the challenges, I would say, of the GDPR-related issues with the collection of photos that have people in them, with the collection of the participants' personal information, our stakeholders, and so on. So some of these were, it's not my previous experience to be part of webinars where we have registration information, for example. That was a completely new aspect. And so what we've done is we've established a physically backed up or based SharePoint, for example, that allows restricted access, is not a cloud-based resource, and so on. And it's just taken some learning and some time to find these solutions, but we've been able to do that in part because we've been working through this planning. I think one of the smartest things that we did is we took on board very early advice about using persistent unique identifiers. So these are identifiers that allow us to go from that moment of sampling all the way through the deposition of the data into the public databases, one name associated with one thing that stays consistent throughout the whole project. I provide here a table that just gives an illustration of how this is one part of how this is described in our data management plan. It actually takes several tables to establish this this naming convention. However, it is much easier to implement than it was to write into the data management planning. But you can see that we have things very well organized and established, and we've used them from the collection point, from the first collection of the first sample. And what I really like about this application is that it is also flexible. So if you take a look at that LU09T1-W1, I can tell you that is a sample from the Luleälven, the watershed in the far north of Sweden. It was the 9th site, and that corresponds through the metadata that is associated with that sample, that corresponds to all of the information about what the site was like. What time, what date, what are the coordinates, and so on are already connected to that. I know that it is a SteriVex filter sample called W1, and then we can add to these identifiers to as we change and do analyses and so on as we go through the project. And so we can really trace all of the information to exactly the right place, the right time, and give it the right meaning and context all the way from the start. One thing that I would really suggest is if you're using photos, make sure that you introduce this right from the beginning, because this is important to keep it in mind. If you have photos with people in them, some way to track that, because memories are short and projects are long. And also perhaps the photo credits. So that's just a small tip. But that's something that has been really helpful to us. And then just in the last few seconds, I would say that the other thing that we did that was really helpful is that we have enforced a standardization of the data format right from the beginning. And that's to avoid one of these problems in the green there, where we have the most time of the data scientists is involved with cleaning and organizing data. So you can avoid having long periods of time in cleaning and organizing the data if you don't allow it to be collected in a messy way in the first place. And so one thing that we've done to combat that is to collect all of the data in a very strictly standardized way through a tool called Kobo Toolbox, where you can only select certain things. That means that only terminology that's always spelled the same, is always the same words, is unified form across groups, across countries, and across times. Only those things can be used. So it standardizes uniformity, it standardizes formats and so on. And we have one major data set, for example, from the start. So we're not trying to patch together different people's data. And I think if you can implement something like that, or that applies to your project, that would be something you would be grateful for later. So thank you very much. These are the members of our data management committee. Some of them are actually in the call today. And you are welcome to have a look at our data management plan. We don't claim that it's perfect, but we do claim that it is being useful to us so far already. Thank you.
Okay, thank you, Jennifer. This was quite incredible presentation, in my opinion. And it seems that you were giving a lots of information and lots of interesting information here. I was really surprised by the high level of diversity in your data. It seems that you are collecting data from many, many places and many kinds of data. And as all the researchers here who are following this presentation, Jennifer said that avoidance of chaos is the word. So you have to avoid the chaos here. So please be careful with your DMPs. It seems that we have here few questions. Could you share the link of your DMP on Zenodo here in the chat? Is it possible, Jennifer?
Yes, of course.
You can share it, yeah. Yeah, and you can put it just in the chat. And someone is, and Sierra is asking, can someone please share the link of the publication shown in the presentation on Fanax and data management plan published on Zenodo? This is probably the same quest. This is meaning the same thing here. Yeah, it's here. And somebody has already put it here. It seems it's here. That is the correct one, right?
Yep, that should be it. And I'm sorry, I see somebody said that it was blurry. I'm sorry, it must be how it was shared. You will have access to these slides, but I'm sorry if that was distracting to you during the presentation.
Yeah, we are also sharing, besides the recording, we are also sharing all the slides. Of course, we will ask from the presentators so that they are shareable slides, but we can share with the people who are participating in this workshop, all those slides. So don't worry, you will get less blurry presentation later. Yeah, it was a little bit blurry, but you could still read it in some level. Okay, I have a question for you, Jennifer. In your project, what was the biggest challenge when you started and started keeping up the data management plan? What was the biggest challenge? I can already know that you might say that you have high diversity of data, and that is the biggest challenge. But what was really the biggest challenge when you started it?
I think it's an important thing to start the work as soon as possible. I think it's tempting to say, oh, we have time for this before something has to happen. And actually time is short. And so I would say actually that getting certain things in place early, really early, was the biggest challenge, actually, getting people to make that a priority when we had so many actions that we had to take in order to go, right? 3 years is a short time and we have a lot to achieve. And so I think it's to some level it was the prioritization, but of course it was also a very steep learning curve. And on a very practical level, I can say that the GDPR things I was not prepared for.
Okay, yeah, this is interesting to hear. So anyone else in the audience, if you have any questions, you can just raise your hand. And
I see David has his hand up.
Okay, yeah, David, please.
Thank you very much. Yeah, Jennifer, also from my side, very impressive talk and very, very tough work that you already prepared. I have data management experience too from an international project. And you said something very, very good, that data management plans can always be adapted, but it is important to have like a starting document that everybody can refer to. I was wondering, this adaptation of the data management plan, to my experience, this basically happens quite often because of necessities. But but how do you keep up with the changes? I know not everybody really approach me and saying I have to change something or so. Is there a dedicated person in your team having this task?
This is a really great question, because it's a hard thing to have a living document because it requires care. It requires food and water, and sunlight and attention. And one thing that has really surprised me is that it's not actually so far been such a big challenge because people see that it's helping them. And so we have an established data management committee that represents, there's one person from each of the partner institutions who's involved in that committee, and is the national responsible person. But I'm really surprised to say this, but it's really taking a life of its own. So we had a consortium meeting yesterday, and there 3 of the early career researchers on their own said, we think we should have a meeting to talk about how we're going to call things in columns and tables that we haven't already organized. So it's really, I think people are seeing that because we have, these interconnections, it will really be in everyone's benefit if we can keep this information organized in a good way. So it's so far kind of happening on its own, but I think it's, something that maybe requires generating that atmosphere of, hey, we can really do this. It really does help us. 5 minutes now saves us 5 hours later. But I don't have a better answer than that. I think there's people who might have more tips and tricks than me on that one.
Well, I think that was a good answer. Thank you very much. It's very, very important, obviously, to keep people motivated and inform them about the benefits. Great, thanks.
Okay, Jennifer, it seems that you have sparked the imagination of DMPs of many researchers in this meeting already now, so that they are already now starting to think what to do with my DMP now and how often do I have to update it.
So yeah, thanks a lot, Jennifer, for your presentation. I think that was really great, and you gave us a lot of idea why DMPs are very worth to have them. And yes, but it's always the same. We know the good things to do, for example, do more sports, eat more healthy food, and so on, but sometimes we don't do. There's also a psychological aspect on this. And yeah, I thought, maybe Sophie, you can share the next slide. Maybe it's a bit like with our data, like, or what do people do with their stuff they don't need anymore? I invite you to a thought experiment. So, of course, there is type A of people. Type A is people, okay, by all stuff, I throw it away or I destroy it because I think no one else can use it or shall use it. So type B of people, they are cautious people and store their stuff for a long time in their cellars because they think, oh, maybe I can use it later, better keep it. And there are people with their old stuff, they like if other people can use it, but they don't want to invest much effort on this, so they just give it to the bulky waste. Or there are people, type D, who really like the idea that their old things are used, so they bring it to the social store. And there's maybe also type E, not a fixed type, but it depends on the personal situation. For example, whether she has time or about the stuff. So that's different types of people. So now I would like to make the transfer to data. If Sophie does the next click for us. So maybe it's also a bit like with open data and fair data, and the next Menti will be to answer the questions. What do you think, your colleagues from the biodiversity researchers, what is about their willingness to provide their data or to make their data fair so that other people can use them? So maybe there are also type A, no open data at all, or maybe people, yes, open data, but only after a long embargo. Or type C, yes, I'm fine with open data, but I don't invest much effort to make my data fair. Or type D, people or researchers that make all the data best as they can in a fair manner. Or maybe also this mixed type of researchers who sometimes behave like type B, and for the other projects or other sorts of data like type D. So Sophie will again, or Harri, copy the Menti in the chat, and you are invited. What do you think? How biodiversity scientists behave?
Okay, the Menti is in the chat. You can go there, and it should work. Yes, you can just think very carefully. We do have some time for this. We have so you can really think about it.
Okay, I think while we are still looking at the Menti, you are all invited if you like. Just comment on it. Have you any comments on that? Maybe what to expect, how it will go in the future, or anything else you would like? Yes, I think, see a hand from David. Please, David.
Thanks, Gaby. So maybe I was a bit surprised, and maybe it's the same reason why Harri was surprised. I expected it to be many, many more the B type or even the A type of answers. Because in the time that I was really involved in science, being a PhD student and so on, it was exactly like this. And now this C option is already, I think, very, very encouraging. And in the future, I hope if people really understand the necessities of collaboration and so on, it might even develop more into the D type. So for me, that's encouraging.
Thanks, David. Maybe, as that would also be interesting for us. So, of course, we hope it will go more to the D direction. Do you think it's more that information is needed for people, or that they researchers need to be convinced about it, that it's useful, or is it more this, time is always short, and they don't want to invest so much to make their data fair? Is there any opinion on that? Yeah, Sinem.
Correct. I have big experience about that. So we are collecting data and we are harmonizing data, and we will see both of you tomorrow, both well. And our experience, why they don't want to share their data, because most of the researchers they are professor, they are old school, so they would like to see if I use my data in proper way. I publish enough with this data, and then I can share, maybe, and there should be an advantage. And if you are in the same sector with these people and you offer a new paper, yes, they may share. But if you don't offer anything and you would like to see it, but it's useful for science, it's a question mark, but how? But my data will be open and somebody can use it as how it is. So it's still not well understood concept. Sharing your data higher the value of your paper and your work, because your work became, let's see, famous, and your effort will be seen in each data repository. People from all over the world can reach to you to speak about your data on your work and future research. And we are still need to invest to maybe this workshops also works very good and to inform the people what is the data sharing and what is the advantage of data sharing. That's why B is still has has votes in the future. I think in 5 years, 10 years, there is no B. I hope.
So let's continue with this workshop and this Menti to see if you are right. So I saw a hand by Heike Felter.
Yeah, I don't know whether you wanted to go on, well, really. So, but yeah, I often experience C actually. So I'll try to be better than C myself. But when we try and check out data from other groups more in population genetic studies, that we download, for example, from Dryad, we very often, it is made publicly available, but we very often have trouble finding our way through tables, Excel files, abbreviations, and so it usually doesn't make too much sense. Or also, sometimes it says in the publication the data will be available on request. Then you request it, and then you get someone, oh, but I have no idea whether you know what you would be talking about if you use my data. You have to show that you have some knowledge before you can work on it. It was like, huh? What's the problem? If I don't have knowledge, I'm not going to do anything with it. So, there's, because the journals force people to make it publicly available, people kind of do it, but they do it in a very sloppy way quite often, or they don't share once you request it, which is kind of sad. And I think many of us, like Selim also said, when you experience the added value of being able to have a large meta-analysis, and then I think, science can make big steps forward. And so it's a pity that people don't share more.
What you said reminded me on the panda video, so maybe you can forward the link to them. Okay, do we have any more comments? I think
it seems that one of the tasks of this DMP workshop is really to convince you to turn from C to B. I think, and I will give my hope for the next presentations here, where we have some open science basic concepts and principles. If we don't have any more questions, I think we can move to the next one. So we have title, Open Science and Open Data, Main Principles and Concepts, and it is presented by Dimitri Brosens, who is a Biodiversity Data Liaison Officer at INBO, Belgium. This is a package of main concepts of open science and data opening. Dimitri has kindly helped us in a tremendous way with his introduction to these things, basic things already in our several previous workshops. So Dimitri, you are working as a Belgian GBIF node manager for your country's biodiversity platform in the Flemish Research Institute for Nature and Forest, and that is your position. So Dimitri, the platform is yours, please.
Hello, everybody. Nice to see you all. My name is, like Harri said, Dimitri or Dimi, and I will try to introduce you a little bit about open science and open data, main principle and concepts. Well, my position is just changed this week. So I'm still in the INBO, but I'm not the node manager of the Belgian Biodiversity GBIF node anymore. I will coordinate in the future Flemish Biodiversity portal, and the hope is that this portal can make use of all the data that you will publish after all your very interesting projects. So I was, until last Friday, member of the Open Science Lab for Biodiversity in INBO, and the goal of our team is actually to make biodiversity research open and reproducible, and we do that through open science. I will not stop doing that, but I have a slightly different position. So we have a living atlas for Flanders in the coming up months or years or days maybe. And I was the Belgian node manager for GBIF. And I think that during this workshop and the next workshops, a lot of GBIF related material will come up. So we'll not focus on that during this presentation. I had a look at the brochure or the Biodiv booklet, and I saw that there were like 3 themes: innovation, harmonization of methods and tools for collection and management of biodiversity, monitoring data, addressing knowledge gaps, biodiversity status dynamics, making use of available biodiversity monitoring data. Well, I think that to achieve this, I think that open science is the way. Open science, open data should be the way, or is the way, and achieving that and getting this map of Europe, which you see in the background, which is GBIF occurrences, even more full, and not only Europe, but the world, of course. Also, in the brochure, I created a word cloud of all the words of the text in the brochure, and the words that are coming up here is biodiversity. Well, that's good, because we are all in biodiversity-related project, data, and monitoring. I didn't find too much words on open science and open data, so I took the liberty to change the word cloud a little bit in the way I would like it to see in the future. So, of course, biodiversity is still the main thing. Monitoring is still very big, but from data, we make it open data. We also have standards, open science, open source, fair data, etc. Tools. So this is how I would like to see things in the future if you make a word cloud of the whole book of the next Biodiversa Plus call. And because I like word clouds, we'll start with another word cloud, and this is the one on open science. And there are some aspects on that: open source, open access, open data, open research data, fair principles, community tools, knowledge, etc. These things were not invented by me, this open science thing, but I think it's actually started in the 17th century, but recently in 2021, it was UNESCO coming with a recommendation on open science. So that's the first very big and large organization advocating for more open science, making science more accessible, inclusive, and equitable for the benefit of all. It's not only UNESCO, I recently noticed that in this open data, we see more and more the movement as open as possible and as closed as necessary. So this is something that is coming up recently. So it means that we have to make as much open as possible, but in some cases, to protect human rights, confidentiality, intellectual property rights, personal information. We all know, or we saw the GDPR law in Europe, that we have to be careful with certain aspects in open data. Also, the Organization of Economic Cooperation and Development also made posts or some publications on open science. And I think that this is one of the reasons here is, if you go back a couple of years to this terrible period of COVID, there was a call globally for scientists to work in an open way on trying to find a vaccine to make this virus a little bit less harmful. And I think this was also a very big push for this open data and open science community. People started to work together, open their data, and managed to get a vaccine in a very, very short time. Also, Europe, open science is very important. There is a European open science policy, and they have on their website, they call it the 8 ambitions of the European open science policy, but I counted 9. So one of them is open data, make your data open, fair, open and fair. There will be infrastructures like European Open Science Clouds, would be a place where you can store, share, process, and reuse research digital objects. They will also, in the future, we know that we have always been looked at to our publications and how good and how big the publications are. Well, they should find new metrics and new indicators to see what our scientific output is. So they make alternative metrics and rewards, and they will also create an open access policy in Europe, people will get rewarded, recognize the diverse outputs, practices, and activities, looking at research integrity and reproducibility of scientific results. Results of research and innovation activities should be reproducible. We'll talk about that later. Also, a point about education and skill. And citizen science is getting more and more important in all this area. Also, I had a look at the Biodiversa Plus ideas about open science. So they fit quite good in all this area. In this area, promotes accessibility and sharing of data. Biodiversa Plus encourages collaborative research, transparency in research processes, public engagement and citizen science funding and policy support, utilization of open access journals and repositories. And there is also a guidance document on the data management, which you probably all have read, open data and the production of data management plans, where we saw a very nice example just before I started to talk. It's not only Biodiversa Plus or the other organizations talking about open science, but also in NASA, they talk about the transformation to open science. In the middle, you see the logo of my forward team, where open science is very important. European initiatives like Open Air, EOSC, FRDN is the Flemish research data network. That's something in Flanders. And CERN, where the collider is also working a lot, the physicists working a lot in the open space. One of the things we'll talk about later is Enodo, which is a thing that came from CERN. Also, in our institute, we do workshops, we sit together, we think how we will transform the institute into an open science institute. The IMBO, as the Research Institute for Nature and Forest, already has a name as an open science institute, but there is still a lot of work to do. For example, we had a potential spearhead, integrated data management and retrieval. So we still have a lot of long-term of old historical data in the institute. How can we mobilize it better and make sure that everybody in the institute can work with it? Can we develop internal data networks? Because we see that between all the teams, it's not always that easy to share and find data. How do we put more effort in the European cooperation to align biodiversity monitoring? I think this is really aligned with the Biodiversity Plus program now. And how do we encourage the application of open science objective in the IMBO? But I'm not here to talk about my institute. I will try to explain you a little bit better or more what is open science. And there is nothing to be afraid of, because open science is something that is actually not that different from traditional science. It just means that you can carry out your research in a more transparent and collaborative way. And it actually can implement these open science ideas in all research disciplines. I would like to make you understand what open science means and why you should care about it. Some of the different ways to go about making your own research more open. Understand also why the funding bodies, and not only Biodiversa Plus, are in support of open science and what their basic requirements are, and be aware of the potential benefits of practicing open science. And that's maybe the most important thing. A lot of this is based on this introductory course. There is a link on the slideshow to the Zenodo package. So the practice of open science is doing research in a way that others can collaborate and contribute easily. Your lab data, your lab notes, other research processes are freely available under the terms that enables reuse of this data, redistribution and reproduction of the research and its underlying data and methods. Opening the research process supports the validation of your data, makes it reproducible, and reduces the cases of academic misconduct. It helps to maximize the impact of your research and provides the foundations for others to build upon. So applying open science in your daily workflow is just a part of good practice. So simply, in a nutshell, open science is transparent and accessible knowledge that is shared and developed through collaborative networks. So I went looking for some information on open science to make a presentation. It's already a presentation which is updated, so this is more than a year ago. But there was a very interesting article, Trusting on the Shoulders of Open Giants, Open Science Increases Trust in Science for the Public and Academics. And there was a DOI there, so a digital object identifier, persistent identifier. I clicked on it and it says you do not have currently the access to this article. So I think there is still a lot to learn. But yeah, okay. As I did not had access to the article, I could not read it completely and promoted it in this presentation. So why open science? Because first, it's good for research. It means that researchers, for all researchers, so also research in developing countries, can see your work. Also, practitioners can easily find your work and apply your findings. And also the public can access your findings. So that's a good thing for research outputs are accessible to all. And the research process can be accelerated and new knowledge can be more quickly generated and built upon to help to solve these grand challenges we are facing right now. It's also good for society because taxpayers get their value for the money. We should all invest in better research, better policies, and better nature conservation. And also, your research has more influence on policy. Open science offers a better return on investment from research funded by public money and contributes to better economic growth. And maybe most important, it is good for you. You make more exposure for your work. And you also gain, and there are articles on this, higher citation rates. And you're multiplying also the number of citable outputs. Think about Zenodo. You can put a lot of stuff already on Zenodo, a lot of things you worked on, make sure that they are citable and that people can, that your citations can be checked. And the last one is that it should be it's compliant with the ground rule. If your funder says that you should do your research in an open and collaborative way, you better do it if you want to get paid for your research. And also Biodiversa Plus is strongly encouraging and asking you, all of you, to do your work in this open science view and way. There are some pillars of open science. The first one is very, this all open data, make your data open and accessible. Open source and code, we'll talk to that a little bit later. Open methodology, open peer review, open access, and open educational resources. Some places are, open access. I think we know this, that you can read your articles and do not have to pay for this. So we'll skip this for now. But another important thing is to share your data. And there are a lot of repositories, places on the internet that can help you in achieving this goal. Share your data. The research data that underpins publication should be accessible to support validation and facilitate reuse. In cases where data sensitivities won't allow open access, be sure to provide details on how someone could request authorized access. And then we are coming a little bit in the area of FAIR. Some examples here, GBIF, people will talk a lot more about GBIF during the next few days. Zenodo, dried figure, come back to that. BOLD system, move bank for animal tracking data, the OBIS or Ocean Biodiversity Information System, the Group on Earth Observation, BCODMO, LTER, Long-Term Ecological Research, and the NCBI, the National Center for Biotechnology Information. So share your data in the places where they should be shared. First one, I quickly want to explain to you, but I guess you all know about this already much more than a couple of years ago. It's a Global Biodiversity Information Facility. It's a place where you can publish, you buy the primary biodiversity data, and the goal of GBIF is to make data access of data free available on the internet. Another one which is very, very close to GBIF is OBIS, the Ocean Biodiversity Information System. While GBIF is dealing with everything on the whole world, OBIS is focusing on the marine realm, but they're using the same standards. It's called Darwin Core, making sure that this data becomes interoperable. And this is also a word that is interesting and a word that we need in this open science environment. There is also a special a place to post your data about for animal tracking data. So many repositories have their own focus. And you are strongly encouraged to publish your data on the most suited repository. eAlter, long-term ecological research. And I think that in most of the pictures in the presentations, there are links to the respective repositories. a lot of results or a lot of ideas about genetic and DNA data, BOLT system, and CBI. And the good thing is that many of these international organizations, data repositories are talking with each other. That's, you can go from BOLT system by one click to GBIF. From GBIF, you can go easily to MoveBank. There will be links between OBIS and GBIF. So this is not pillars. These are things which are trying to work together and trying to cooperate to build something great, which is already happening now in the future. So I talked about specific repositories for biodiversity data. You go to GBIF, but what if you have data which is not very suitable for a specific repository? You can go, for example, to Zenodo. This is free, and you make an account and you can actually post a package of data. And there's several ways of posting your packages of data. You can also provide metadata, explain what is in your package, so people, when they download it, can at least understand for a big part where the data is about. Think about the panda. The guy didn't do anything. So you explain in your metadata where the data is about. This is Zenodo. Also in Zenodo, and this is maybe interesting for you, you can easily create communities. And I did not create this one, but this is a community from the INBO, the Research Institute for Nature and Forest, and all our publications on Zenodo will be indexed in this repository, in this community. So it could be possible to create a community for all the Biodiversa plus Biodifmon projects, or you can also create a community for your own project and get all the stuff, all the images, all the posters, all the non-peer-reviewed articles, even the peer-reviewed, you can post them there in this community. And that makes it very easy to find this stuff back. Dryit, it's more similar than Zenodo, but mostly aligned with, if you make a publication, the data which was used to come to your results might be stored or published in Dryit, which is not completely free. Figshare is also a similar one, a data repository on the internet. Well, we're talking a lot about open data, but of course there's it mentioned before, we're also looking a lot more to fair data. And fair data is data that is very similar to open data, but it's not completely the same. Open data can be simply if you put an Excel on a website which is freely available and downloadable by anybody, anyhow, you can call your data open data. But what is the advantage of just putting a data file somewhere on the internet? You should make sure that your data can be used, can be reused, can be shared again, and can contribute to a scientific effort. So something was invented already in 2016, I think, and it's called fair data. Make your data findable. Persistent identifiers. We've been talking already about DOI, digital object identifiers. These are identifiers which you can click on the internet and will always lead you to the same data package or the same package. Make sure that your data has sufficient metadata explaining what's in there so people can understand. They will not misuse your data or make wrong interpretations. Follow naming conventions, use keywords to make stuff findable, and versioning. Make sure it's accessible, and there is a little bit of difference between open data. In fair data, data can be in a data repository which is protected and not that easily accessible, but it should be findable. People can ask you, okay, I have the metadata, this is something interesting, can I use your data? And you can come in contact with the people who own the data, and they might send you a version of that. Make sure that the data is interoperable, use as much standards as we can. In our biodiversity world, we mostly, as far as I know, mostly rely on the Darwin Core standard or ABCD standard. These are standards making sure that you can take one data set, another data set, and can easily merge them in a more complete data set. Be aware you still, as a scientist, have to check if the data is really what you need. And GBIF and all the other data sets and repositories are not without mistakes. So be vigilant for stuff. Use similar vocabularies, methodology, and references. And to make the data reusable, put the proper license on it. Use community standards. A small cartoon on fair data. There are zillions of cartoons, but I like this one. Findable, accessible, interoperable, and reusable. There are tools on the internet to check if your data set is fair. This is a kind of a wizard, and there's also a link in the presentation. So you can just complete your, you have your data set, complete the wizard, and it will give you an indication on how fair your data is. This is a small diagram on the differences between fair and open data. I think the most important difference is that for fair, it needs to be, it doesn't need to be open, but it needs to be findable. And open data is not always findable. But I think that we can say that fair and open data would be the best solution in the future. Another aspect of this open science is you cannot only publish open access, share your data, but also share your code. And there are several initiatives, and the most important one at the moment is GitHub. GitHub, I call it sometimes a Facebook for nerds, where people can post their codes, explain their code, and make sure that their code is reusable. For us, we have a lot of repositories on data publication, and what we do is we have SQL queries what we create, we have RScript what we created, and we all have them available on GitHub. These are mostly R scripts, so people can go there, reuse parts of the script, and it's simple. It can be just changing the coordinates from one coordinate system to another coordinate system or whatever, making connection with GBIF, certain different scripts to map stuff to DOM record. That's a little bit Oriel, but making sure these things are available, the scripts are available first, make sure that people can reuse it. And also in the open science area is that if people get your data, look at your scripts, they can rerun your research and actually check and see and believe maybe better your results or understand better your results. So not only open access to publications, share your workflows and methodology. An interesting website for this is the Open Science Framework. This is a website where you can use it as a project website where you can post a lot of issues, and they're mostly, not all of them, are open for everybody to see. We use this first for one of our first open science projects, which is called Tracking Invasive Alien Species, and that was already in 2017, which is a long time ago. Another aspect is open peer review. We also want better research. We want good review, and there's a kind of movement making the review process more open. So that means that if you author an article, you will know who your reviewer is, and you can have a better discussion, a more open discussion on improving your research. So making the peer review process more transparent, researchers have better access to the peer feedback at an earlier stage in the life cycle of their research. And then open educational resources. What we also do, we have something in IMBO, which is called IMBO tutorials. And there's a lot of stuff, a lot of code, a lot of tools, what we use for data manipulation, research, that we put open out there on GitHub. It's all hosted on GitHub, so other people can use that. And it's been used because I was on a workshop some months ago in Croatia, and I saw people. I was talking about these IMBO tutorials, and people actually know these tutorials. So actually, I never know these people, saw these people, but they knew the tutorials. So we are having the idea that we're not doing this for nothing, and we like the idea that our stuff is being reused. Of course, as scientists, we like to have taxonomies. So this is a open science taxonomy. A lot of words will not go into every one of these, but I think I try to highlight the most important ones. And I got in the last part, some ideas for opening up. So you're already been granted the project, so the planning stage is already finished, but for in the future, important, find the relevant research question. I guess you all did that. Make your research question sharp and clear, and make sure that your research is relevant with the research question. Seek involvement from other researchers. Be sure to consider involving other stakeholders too. Check already some data repositories to see if there is existing data that you can already use or build upon. We mentioned this before, or this has been mentioned before, write a data management plan. It's a plan which is, like discussed before, it's changeable. You can change it over your research cycle, but it would be nice to have a good plan to start with. Also think about writing a communication plan and foresee revision, and also already during the planning stage. During the active stage, you have data collection. So make sure that you get your data through well-defined protocols, also mentioned in the data management plan in the first presentation. Make data collection automatic if it's possible. Existing data is reused when possible. Make sure that you have good validation procedures, and understand your measurement biases and document them. You cannot just go and download data, use it, put it in an R script, do the analysis. No, check your data and get rid of the stuff you don't need. Also think about data storage. You can make a data management plan, and then if you have it, and sometimes you need to make it to get the grants for this one, for example, you can make your plan and just put it aside and don't look at it anymore. In a certain moment in your research project, you will hope that you looked at your data management plan. I'm sure of that. Metadata, provide metadata, and of course be aware of sensitivity. This is the most difficult word for me. Sensitivities and sensitive data. Also, make sure that your data validation is reproducible. Make fixed validation rules. Make a scripted data workflow. Make sure your data analysis is adequate and correct. And your analyzed workflow is open and reproducible. Make a versioning system that if you change stuff, that this is noted. Code and script are keys. Are kept. Sorry. Then during the active stage, you might share your methodologies already early via preprints. And are you worried about getting scooped? Or if you share early, pre-registering your studies give you time-stamped evidence of your ideas. In addition, any peers that review your early work and vouch for you, the Open Science Framework, that website I mentioned before, offers great advice on pre-registering your project. And towards the end of your...
Sorry, 1 minute.
1 minute. Towards the end of your research data sharing, publish your data as open, make sure that fair principles are followed, use scripts and open your source code or your code and open protocols. Publish your stuff as open access, use correct citations of resources as sources, and use an org ID for yourself. This is a specific ID for you. Make sure that all the documents are achieved sustainable, open in an efficient way. Archive your data, archive your samples, archive your protocols to make sure they can be reused, and archive your source code. And create opportunities for internal and external use of all researchers, and make somebody of your team open science champion, someone who is interested taking the lead on this. And very quickly, the research circle changed from just ideas with this data collection, analysis, visualization, interpretation, and publication to something a little bit more extensive, where you will put in new data, existing data, get it from GBIF Zenodo or another repository, use the data, document, and you see that the research cycle got a little bit bigger, but it will be helpful in future projects and doing your work in an open science way. Voila. This is more or less what I wanted to say. If there is time for questions, I'll be happy to try to answer.
Thank you, Dim, very much. Thank you very much. Just very shortly, you have shared with us so much useful information that it will be really useful to share those presentations also afterwards. We have a question from Sylvain, please.
Yes, hello. Thank you for the presentation. It is more of a comment rather than a question. I am one of these people who put like a stone on top of my head in the beginning, the first ice-breaking thing. And I think that this stone is very heavy right now, like after presenting all this. You should do this, you should do that, you should do blah, blah, blah. Like, I am an ecologist, right? Already, I've been asked from the projects to involve stakeholders. I don't have a training for. I'm trying to be trained for this. It's very nice. I see the value, and I completely see the value of having open data and fair and all this. It's just that I'm wondering, how much time in the end I invest in this, because in the end, the benefit is for the community, and it's fantastic. I really like this and I support this. As we were saying before, like a lot of scientists keep their data for them because they want like this to have more publications and to be on even scientists that are talking about open science and all that. I know personally people that talk like this, but then when you ask for the data, they want to be part of publications in any case. So I see really the value. It's just that I am an ecologist, and what I am asked to do is much above, I think, my current competencies. And of course, I can try to do some things, but I don't know, like time-wise, how does this translate? This is my question, and maybe Jennifer can contribute also on this because she did this great presentation before as well, and maybe she has her own experience that would be helpful also on this. Thank you.
Okay, thank you for this comment or question. Well, I'm lucky in the position to have in a situation that we know people that can help us or that are helping us with this. It is definitely something where I do not want to have the stone and give the stone to you and make sure that we have, like I said, we have in our institute, we have an evolution to an open science, and there are people, we have data stewards and people that can help researchers in this process. I think for me, maybe a lot of the stuff is logical because I'm I'm looking at that already for a long time. But if it's all new, it's something new. One important thing, I said it in the beginning, it's not so different as in open, as conventional research. You make a script, you probably have scripts. Well, in this open science idea, you make your script. It will take a little bit longer, but also for yourself to use it. You document your scripts, and maybe not just R, but the R Markdown, where every step you explain to yourself. And if you come back after 2 years and read your script, you will still understand it. And I'm pretty sure if you just write your code and do your analysis after a couple of years coming back, it won't be that easy to understand. So I think people should be around you to help you because it's not something you should carry on yourself, and you do it also for yourself. So making this research in an open way will help you also to understand what you did in 5 or 10 years.
It's just that not everyone has this help. So this is something that Biodiversa and generally, like you should understand that not all institutions have the same means and the same support to their employees. So like, and I am in Italy, I work in Italy, and we don't even have funds right now, right? We haven't been funded yet. And I know that like in my case, my institution doesn't have this. So for these institutions, maybe like Biodiversa can support give some extra support, or should have less expectations. I don't know, but somehow it has to be, yes, differentiate.
Okay, 10 short comments from Jennifer and Heike. Please, thanks. Jennifer first.
Oh, okay,
Yeah.
thank you. I just want to say I'm an evolutionary biologist and I know how you feel. This is outside of my comfort zone. There are not really stakeholders for evolutionary biology, even though we think you should all be considered that. So I understand the feeling of being overwhelmed very well. And I think one thing to think about when you think about that stone on your head is that it's not just your hands and your head who have to carry that weight. And so I really think one of the smart things that we did was we made a committee so that your different partner institutions have an active role in helping to carry the weight of that. And they might also have access to resources that you don't. You know, we have partners who come from a diversity of institutional situations, funding situations. You know, we have Italian partner as well. You know, I think it's remember that you're a team in this. Yeah, start with maybe, you know, some optimism, even though I know that it can feel really, really heavy.
Okay, thank you. Then Heike, please.
Yeah, I just would like to comment on one thing in what you said is for what we potentially eventually also need to discuss within Biodiversa or on EU level. And that's that we quite often equate open access publishing with high quality publishing. And that's, to me, two very, very different pair of shoes sometimes. And, you know, we have this discussion in, so I'm evaluating in the Leibniz Association in Germany, where quite a lot of taxpayers' money is also placed, not only DFT, and where all the institutes are urged, you know, publish open access, and then there's really this thing like, okay, now we have a much higher quota or ratio of open access publications, when in the end, you know, you look at the publishing organs or the journals and you think, maybe not a good choice. And I don't know, I don't want to go further into this because this can take the whole afternoon potentially, but I think this is probably something we should keep in mind for future discussions when it comes to open science.
Okay, thank you, Harri. I think we have to move on because we have one Menti, but we have also, even more important, we have a break which is coming soon here. So thank you so much, Dimitri. This was truly interesting, and I hope that it was also a very inspiring speech you gave. So now we are going to have one very light Menti here, and then we will let you for a break. Okay, yes, I think you can see it now. So let's have a vote again. This Menti will be about the source of the data that you have used during last 5 years. Where does it come from? Is it from your own data collection, data from the people you are working with, or data from repositories, GBIF, etc.? See, and it seems that the own data collection is winning. So we have a little bit over 20 minutes time for a break. So everybody needs a biological break, but also coffee, tea, whatever you are using. So see you again in more than 20 minutes. Okay, let's continue. I hope that you had a relaxing break. Now we can have the two last presentations in a row. Both of the speakers are representing GBIF. And as a first speaker, we have here Birgit Gemeinholzer, who is a GBIF Science Committee Chair. She will give a presentation about data management and open data in GBIF. Global biodiversity data sources are truly in a very important role for Biodiversa, but also in collecting and sharing species data. Birgit, you have this kind of a double role now because you're a speaker, but you are also a funded consortium leader. And besides this work, you also work as a group leader at University of Kassel with metabar coding and population genetic analysis of endangered plants and biological data accessibility. Please, the stage is yours, Birgit.
Thank you, Harri. And yes, I have many hats on my head, and I would like to talk to the people who feel like a brick on their head. So the other two talks were so good. I'm really impressed about Jennifer's and Dimi's talk, and I will talk to the people who say, well, I'm not very familiar with data management and open data, and I want to show you how you can get a foot into the door, and I hope that at the end of the talk you will be convinced it's worth trying to open the door and push it a little bit. Oh, sorry, my doesn't work. Yes. If you think about data management, often it's only about I have to do a data management plan, I have to do something, but it's not only that you have to do something, but you do get a benefit from that. If you think about data literacy, to be data literate, you know things about reading data, what's the theory behind it, what's the analysis behind it. You knows more about communicating with data. So the reporting and the presentation of data and working with data in the collection and management possibilities. And of course, all this combines data literacy, and at the end, this is the benefit for all. But integrating research to data management into research and teaching would be the first step, that everybody just learns it from the start, and everybody would use it and say, well, it's easy, it's simple. Getting into that in a later stage and thinking about how do I get it organized is more difficult and more complicated. But I think we have to work on that, and that's why I think it's an important topic. And I hope at the end of this talk you will think that also, even you think, well, I have a brick at my back. There's a real data value chain. You all spend money which is publicly funded, and it's a lot of money we get, which is a great thing because we can do great things with that money. But with the data creation and the data acquiring, we also have to have a data transformation. We combine, transform, analyze, and then we hope that our data will have an impact and will influence decision making. And this is really beneficial also for others. There's a need, a user chain, a value chain by users, communities, business, markets, social science, and so on. And the real world data will go back into the creation of new data. And this is really beneficial if this data is available and can be built upon the data, if that's not a standalone data set you have. So, well, you might think, who is interested in my data? It's only my data. We are interested. And it's not only one person being interested, but it's a community that's interested in your data. And I hope I can convince you that this is really worth doing all the effort and all the work. But of course, I do have grumblers that tell me, oh, why should I make my primary research data accessible? It is boring. And you heard a lot of arguments before why we think it's important. You think, well, what's the benefit of it? And I will want to show you that there is a benefit for that. Of course, the grumblers say it takes so much time to reformat data prior to submission, and that's correct. But if you do it right at the start of your analysis, if you use an Excel sheet that's really compatible with all the different standards, it is so easy. So do it right from the beginning. This was also Jennifer said before. My last grumbler, of course, says, oh, I don't know of any archive in my field of research. I don't know who can help me. I don't want to do it. But of course, there's a carrot and there's a stick. And I do like that Biodiversa is really supporting us and a little bit providing us the stick, but it's also the carrot that is in front of us, and we have to use the carrot. And I want to convince you that the carrot is really a nice carrot. If you look at publications, there are several publications out that look at the value of research data. And there's a citation analysis of the oceanographic data sets, and they said that the estimate citation counts are 99% higher of all the journal articles because the data is open access. So there really is a benefit, and it is seen in the community that the data is available and you can build upon that data. But also if you say, well, I have another topic here, the data publication correlates with citation impact, and that's really true that on average the publications that have open access related tags achieve a significantly larger citation impact than the average in the field. Another plus article, the citation advantage of linking publications to research data, have a statement that in the link to data in a repository are cited 25% more often on average than normal data sets. So it really is beneficial for you and for everybody else because they can build upon your data and add data to that. And you already saw that from Dimitri before, that there's this data life cycle and you plan it at the moment. And if you plan it right, you go out and collect your data, you make assurances that the data is good and well described. And then you submit the data and it will be preserved. Somebody can use it to discover something, integrate it, analyze it, publish it, and then somebody else can use a plan to define a new study. And the good thing about this data life cycle, what it's all about is that your part is only that one, and others will help you and support you with all the other duties, and you will benefit from that. So and don't be worried too much that you have to do so much work. There's so much in the background other people are doing for you to link data and make data accessible. And it's really worth the effort to do this part of the data life cycle. Well, you say, well, it's too complicated. I can't do it. There's so many initiatives, and most of them are at the moment on a national scale. So I here present a few. One of these, the Swedish Biodiversity Data Infrastructure, the Norwegian Biodiversity Information Center. There's a Dutch National Dashboard for Biodiversity, and you can contact them, their help centers. The German one is establishing currently as NFDI for biodiversity. I learned tomorrow you will have some insights into the Portuguese biodiversity research data infrastructure and so on. So there are emerging bioinformatic data centers that help you how to do it best. And there are also these world repositories like Pangea, where you can really deposit your data, and it's big, findable and accessible, so it's fair data. The genomic community is an easy community because they are doing this data storage for such a long time. The International Nucleotide Sequence Database Collections, the INA, the NCBI, or DDBJ, they used to work with open data. And if you want to publish in the genomic community, you always are used that you have to submit your raw data before you publish. This is not the case in other fields of research, and of course it makes it more complicated, but I think we can learn from them. Here, you just have to do it. You know before you publish, you have to submit your raw data, and we have to do that with all the other scientific publications out. No publication without primary data submission would be the way to go. And of course, you can ban it for a certain time, so it's not open access just right at the beginning, but it is in the review process and it's available. We know that there's so much more data than only the genomic data, and you have the distribution models, the ecosystem data, the experiments, the functional traits, the collection data, molecular data, model output, satellite data, occurrence data. Before there were people asking about evolutionary genetics and so on. There's so many different kind of data out there. But especially the national data centers, they will help you and they will serve for that, and they can support you in finding the right repository and knowing what to do and how to best store your data. To my knowledge, these biodiversity information data centers are now establishing tables, prepare Excel sheets, so you can use them right away and make easy submissions. So the tools are developing, so keep an eye on that. You are not alone out there. There's a whole big community out there that's willing to share data, and being willing is the first step to go on the right direction. At the end, we would love to have all the organismic biodiversity data in GBIF. That's why I'm really here with my second head on the top of the GBIF, the Global Biodiversity Information Facility. It's an intergovernmental network in research infrastructure funded by the world's governments. And this is where all the biodiversity data should go together because it's free, open access available, free to use, and it really gives information about where all type of life on Earth is, and everybody on the whole globe has access to GBIF data. And GBIF is really the driving force. It's a network of networks of networks, so really merging biodiversity data with a lot of training and support and help and technical expertise and many, many people being there to really keep the network running. But this is also the benefit that everybody can use the data wherever you are on the globe. You can have access to this data. There are these voluntary collaborations through memorandums of understanding, so supported by the participant notes and secretariat in the Copenhagen and Denmark. If you say, well, how do I do that? That's the way to go and see how they do that. And currently GBIF holds approximately 2.9 million occurrence records of biodiversity occurrences across the globe. The data comes from 105,000 data sets on more than 2,233 publishing institutions provide data to GBIF, and a lot of data papers have been stated that they use GBIF data, and currently there are more than 10,000 peer-reviewed papers using GBIF data. So if your data is also on GBIF, you might also be cited. So that's really an important thing for a researcher. GBIF is really a window of evidences where species have lived and when. And of course, GBIF started with museum collections, herbaria, and zoological collections, but now they're emerging and they look at observations, at satellite images, at DNA-based work, and so on. And GBIF currently is in the stage of taking more data than only where, when, how, who, and where, when, how, what, and why, the 5 questions GBIF wanted to ask at the beginning. But now it says, well, I can take other data formats, I can take other data files, and I will want to show you what GBIF is currently doing. So GBIF really provides data in very different fields to support research and sustainable development in conservation, in protected areas, threatened species, invasive species, and so on, towards food security, climate change, human health, and so on. These are topics that are relevant for our Biodiversa project as well, especially doing monitoring things. GBIF is a collaborating community of practice. You have a lot of people. They provide open access tools and guidance, training and mentoring. There are national nodes, as I always said, it's a node of a node system, and node staff and partners share skills within countries and across continents, and they have funded projects for capacity enhancement and outreach activities. So if you want to get a footstep into the door, have a look at the GBIF website. There's a lot of information there already to get started, and maybe to get a contact point in your country to see how to proceed. We have this Biodiversa funded project, Meta Plant Code, and we're doing plant meta barcoding, and it's a big consortium, as I showed you, and I want to show you how we can use GBIF as a resource for our data. And we go out sampling in the field. We have our laboratory chains and the biodiversity analysis, and of course we want to make predictions about climate change, time changes, phenological changes, invasiveness or occurrences, and so on. And we can do that with meta barcoding, of course, quite nicely, looking at plant traces in feces, in alcohol, in malaise traps, or in wind spore traps, or in the soil. And we now can publish our data in GBIF, and I want to show you a prototype. And GBIF asked for an invitation, has an invitation open to share DNA meta barcoding data in an early pilot of data publishing tool. And if you put your data into that, you find the dots in GBIF, and if you click on the dots, you find your species list of a meta barcoding analysis. And this is being possible because GBIF started with a new data model. So not only the easy questions of observations are visible in GBIF, but you really can use many more pipelines and documentations and standards and so on. So if you do work with meta barcoding, a lot of groups do, as I know, look at the publishing tool in GBIF. It's really nice to work with, and you see that the trap data is here all over, and you get the species list in a GBIF context with species name. And if you are interested in what's happening here, look at the GBIF pages of eDNA-derived data, the guidelines of publishing, invitations to share meta barcoding data. There's outreach campaigns and so on. So have a look at GBIF at the web pages, and it's quite nice to scroll through the different pages. Well, you say, I'm not working with eDNA data or meta barcoding data. I'm not interested in that. But the new data model of GBIF is also investigating other data types at the moment. GBIF develops standards in an international consortium, and the standards have been ratified. It's called the Humboldt extension. And now GBIF asks pilot projects to see if there's some missing terms, if everything is in place. So at the moment, they look at the collected observations of bird with eBird volunteers. They look at the field museum rapid inventory data, if all the fields are there, so the field museum can push the data to GBIF. They look at squid and fish in pelagic zones of the Cosmonaut Sea and Price Bay. And they look at hummingbirds in northern Andes, and so on and so on. So they try to see if other data types also can be pushed into GBIF, like it can be done with a pilot study of the metabarcoding data. So this is the place to have a look out for the future to see, well, this is the standard development, and then we can link into that and add our data as well. So with that, GBIF really is a window of evidence about where species have lived and when, and we want our data to be there as well in an open and free way to be used by everybody. So with that, I hope I could convince you that you don't think anymore that your research data is not of value for anybody and nobody's interested in it. There are a lot of people out there who want your data, and I'm very thankful for your interest, and I'm open for any questions you have.
Okay, thank you very much, Birgit. Wonderful presentation once again. Yeah, I have to say again that this was really interesting again to see that there are so many data providers nowadays, and when what comes to GBIF, you have given some impressive numbers. Like I really cannot believe that you have over 10,000 peer-reviewed papers with GBIF data, which is kind of a really big achievement which you have done during some months ago. Yeah, I think we have here. I saw a hand somewhere. Somebody. And there is also in the chat, there is a one question. What if my research data is mostly subsampling of existing data sets or simulated data? How to archive them?
Yeah.
So
GBIF is mainly interested in primary data. So GBIF would not be the right repository for codes or models, but Dimitri showed some repositories like Zenodo or GitHub that would be the right places where you remodel, restructure these data sets.
Okay, yes. I hope that this really answered your question there. So, anybody else? I think I saw one hand somewhere.
I could also illustrate it further because GBIF gives you a DOI. When you download a dataset from GBIF, you get a DOI. And if you merge different datasets, you can just use the DOI so anybody else later can redo your analysis. So you mention the different DOIs you downloaded from different repositories. You use your code and you make a code open access available. And then everybody in the future can go back and trace back with this data set. You can, and this code, you get the same modeling in the future. Of course, the data might change in the future, so you might add more information to that. And by using the same code again with more data, you might get different results. But this is a good scientific practice use, so we really should follow that. And GBIF is supporting you in that, providing DOIs when you download a data set there.
Okay, yes, yeah, thank you. I have also one question or some kind of a comment. Well, I have understood that the GBIF is also active with IPBES, especially if you think about the latest invasive species assessment from last year, I think so. So you are kind of creating also this global impact in biodiversity protection, and also with International Union for Conservation of Nature, EUCN, they have also a database of global data sources. Are you also cooperating with them because they have listed like almost 200 global data sources?
Yes.
Biodiverse data.
In the IPBES report, there are several places where GBIF is mentioned, and there's a close link and close cooperation also with IUCN, CPD, and several other policy-making bodies. There's a close linkage with that, and we hope that, always there's a mutual benefit that sharing the news about GBIF and also making use of GBIF data is that both sides benefit from GBIF being there. And that's the same for you. Providing your data and getting data back should be the benefit you have by looking at GBIF data.
Okay, great. So this is truly impressive work on a global scale. And the global scale is now bringing me to our next speaker, who is also from GBIF. We have Melissa Liu.
Harri, I also see a hand by Isabel.
Okay.
Yeah,
Isabel, please.
thank you. If I can quickly, can you hear me? Yeah, yes.
If you can speak a little bit louder.
Yes, if I can just make a quick question. And it's connected to the data which, for which we cannot get to a name. So when we are using more and more molecular data and we get only OTUs out of it, how does it work for GBIF? Because depending on the data sets, we might have OTU 1, 1, 2, 3, 4, and then another person will have a different one. So how can we then homogenize that at GBIF to make sure that people benefit looking this sequence that is exactly the same one from a different work? Thank you.
At the moment, there's a huge project called Checklist Bank, which is trying to compile names on the globe, synonyms, and so on, and see how they are traced back and how they are connected. And of course, if there's a sequence which doesn't have a name yet, it might get a name in the future. So there might be a high likelihood, if this information is available, if the dot is available, the current state is available, that this dot having no name at this time of the year, just today, it can be giving a name in the future. And I think that's the benefit of not only starting a new database, a new system, but adding to a big system on the long run.
Thank you so much.
Okay, thank you. Then there is a long question. I do not know where it is referring. It is, can you put the name of this huge project in the chat? I didn't understand the name. What are you referring to? And Andy, you can also ask.
Yeah, that was a question to Birgit. She mentioned this EU project that could give some examples to the question that Isabel has just made, but I didn't get the name. So if you could add the name or the link or both into the chat, that would be helpful.
I put it in the chat, gbif.org.
Aha, well, this I knew, yep. Okay, thank you.
Okay, right on. Okay, any more hands or questions or comments? We still have 4 minutes. We are ahead of schedule at the moment. But we can also start with the next presentation. Okay, I don't see any hands. So now I will continue with the presentation of the next speaker, Melissa Liu, also from GBIF. Her talk will be about introduction to GBIF training package on data mobilization. Melissa gave the same presentation also last year because Biodiversa, together with GBIF, we have been planning to give this kind of a training course, which will probably take place during next year, during Q1 or Q2, probably during Q2, but more about that a little bit later with my final words. So, GBIF is organizing these special training packages for biodiversity researchers, and what kind of help researchers can get from these training packages. And we have asked here Melissa to present the basics of those training courses here. So Melissa, you are bringing this global dimension here as you are working in the GBIF Secretariat of GBIF Asia support team, and previously you have worked at TaiBIF, which is the facilitator of biodiversity open data in Taiwan. So please, the stage is yours, Melissa.
Thank you, Harri. I think you can see my share screen now.
Yes, perfectly.
Okay. And hi, I'm Melissa Liu, and the member, what Harri mentioned, the member of the GBIF Asia Regional Support Team, and also the member of the GBIF node in Taiwan. And I think my presentation is very different from the others. So I'm going to talk about how GBIF has developed the capacity enhancement activities and take the Asia experience for an example. Oops. Okay. And the works we do at GBIF is guided by a 5-year strategy framework in the 2023 and 2020 to 2027 plan. The third priority area has been to enabling the network to meet future needs and challenges. This recognizes the fact that GBIF is not only focused on delivering data for use, but also on a network of people, institutions, and countries. This network of people has the largest responsibility for supplying data to GBIF, and it is very important for GBIF to achieve its goals so that we contribute to developing capacity within this network of people. Broadly, what we are trying to achieve with capacity development in GBIF falls under 2 main objectives. The first is to develop the capacity to mobilize biodiversity data through GBIF, which includes supporting biodiversity data management and quality. And the second one is to develop the capacity to use GBIF-mediated data both in scientific research and decision-making at all levels. We target these objectives by working to develop capacity on nested levels that you see here, from individual level to regional level, which all contribute to our global GBIF network. But in today's presentation, I will basically focus on the individual level. The goal of the individual level is to strengthen and grow GBIF's community of practice. And when we talk about community of practice, we mean the individuals with the skills, knowledge, and motivation to share and reuse biodiversity data for conservation and sustainable development, and to support others are participating in the community. GBIF has a very strong volunteer community of ambassadors, mentors, trainers, and translators. We are lucky that in our biodiversity informatics community that so many individuals are willing to come together to learn, share, and pay it forward. The GBIF Secretariat, in collaboration with our community of trainers and mentors, have developed short course curricula on biodiversity data mobilization and data use. These courses are available online and for self-paced learning, or can be adapted for on-site or virtual workshops. GBIF has organized so far 15 training events under the 2 programs. One is BIFA program and one is BIT program. And for over 370 people. As a direct follow-up, projects funded under these 2 programs organized over 130 replication workshops that trained over nearly 4,000 people. In addition, data mobilized through GBIF projects has have already been cited in nearly 1,700 peer-reviewed papers. GBIF has organized virtual workshop for project teams instead of on-site ones since the COVID-19 pandemic. So the Asia Regional Support Team has self-organized an annual virtual workshop since 2022 that's open to all data holders in GBIF Asia community, not only for GBIF funded projects. And so far we have received more than 300 responses that say they are interested in attending a data mobilization workshop, and there were 60 virtual and 27 self-paced participants attended in the past 2 years. And there are 14 datasets published to GBIF with more than 2,000 occurrence records within a week after the workshop. We have a small competition after the workshop, and 12 new publishers, GBIF publishers, were endorsed. And next, I'm going to talk about me and my Asia supporting partners' experience on how we organize virtual workshops for Asia in 2022 and 2023. From the presentation agenda to post-workshop activities. Since 2017, with the program BIFA project I just mentioned in the previous slide, funded by Japan, each year, GBIF arranges a data mobilization workshop in Asia, targeting BIFA project teams. So these workshops equip regional research groups with the fundamental understandings and practical tools for project planning, data capture, data quality, and data publishing. However, Japan no longer supports the funding, so there were no workshop plan and target project participants in 2022. It was a challenge, but good chance to engage a wider community beyond BIFA. We started with an interest survey to know the willingness of workshop participants, and then decided how and when to organize a workshop. And then designed an application form and spread it out to potential communities. Then we set criteria and filter participants. Meanwhile, we contacted previous workshop mentors to recruit a group for mentoring participants during online sessions. So let's go deeper to see the workflow step by step. And in the interest survey part, we created a Google form with several questions such as the willingness of publishing data to GBIF, the purpose of attending the workshop, etc. We send the form to the potential communities, especially new data publishers and some institutions we have made contacts, and also ask participants in Asia Office Hour. By the way, the Asia Office Hour is an online 1-hour open QA session for Asia on every Thursday. So after receiving responses from the survey, we decided how many days and the workshop agenda based on the previous BIFA training modules, and discussed the suitable workshop days among our team. And then the most important thing, announcement time. We promoted the workshop through several channels. The first is sending emails to inform the respondents from the interest survey. And second, we asked the GBIF Secretariat to send emails to the Asia nodes, and also created an event page on the GBIF website. And last, we promoted it during the Asia Office Hour and other social media such as Twitter, Facebook group, because it's like popular social media in some Asia countries. So we basically use the Facebook group. Like, since we received the applications more than our expectation, and we only allow 30 participants for the learning quality and efficiency, we set the criteria to filter if the applicant has data sets to publish soon, is a data holder or work with data holding institution, has never participated in related workshops, or plays an important role from nodes or can fill the regional gap, or plan to hold replication workshops, or wants to become a mentor, even a trainer, and support the future workshops. And on the other hand, we recruited a mentor group by sending emails to the GBIF mentors and assign them to different participant groups based on the country or the languages they speak. Here are the resources provided by GBIF training team that could help you prepare for the workshop. The first is the biodiversity data mobilization course. This is for participants to learn in self-paced and for trainers to refer to the rubrics of each assignment. And the second is the virtual workshop planning notes. It is for organizers to know how to organize a workshop and get all the information needed. The third is GitHub, the trainers to download all the educational resource from. The materials are also translated in multiple languages. If you are interested in joining the translator community, just please contact the GBIF training team or we Asia Regional Support Team. So the workshop module is actually based on the GBIF Biodiversity Data Mobilization course and has the 3 phases of activities. The first week is self-paced learning and material review, and the second week is the 4 hours Zoom sessions for 5 days and part of online self-paced learning. The last week is the exercise submission, and then the trainers will do the grading. The length of the workshop is flexible for different needs and purposes. You can also set up a half-day workshop for the basic introduction, or a 1 to 2 days workshop for the topics you are currently keen to know. In the grading part, the trainers need to review all the exercises submitted by the participants, and it takes a long time. And mostly refer to the rubrics of each topic and grade the exercises. And after done the grading, all the trainers should write down the scores in the sheet and see who can get the badge. If the participant gets the average score above 2.5, they can earn a basic badge. If they get the average score above 3, then they can earn an advanced badge. The benefits of badges are that you can know the skill level of data publishers and provide good support and communicate with them more efficiently. Also, it is the basic criteria of becoming a GBIF mentor. We also try to encourage participants to publish data sets to GBIF after the workshop. If they publish a data set within a week after the workshop, they can get some GBIF goodies. The GBIF Secretariat welcomes GBIF nodes and partners to apply for the goodies for workshop incentives. So this course would not be possible without the developers of this course and the trainers and mentors that serve as instructors and group leaders to facilitate learning and provide ongoing hands-on support to participants. Some of the workshop participants even became GBIF mentors and trainers after earning the badges. So as you can see, developing capacity at the individual level contributes the empowering people and increasing data literacy and growth in our community of practice in turn, like resulting in a growing new number of open data ambassadors, mentors, trainers, and translators. So I'd additionally like to take the opportunity to thank all the authors of this course, Laura, Maliani, Mahiva, David, Sharon, and Sophie, along with those primarily responsible for the design and instruction of this course. And if you are interested in reaching out to the Asia Regional Support Team, you can email us or subscribe to GBIF Asia mailing list, or join our Asia office hours every Thursday. It's in the afternoon in Asia, but I think it's in the morning in Europe. So you can also discuss any data issue on GBIF Discourse forum. Okay, thank you.
Okay, thank you, Melissa. Thanks for a very clear presentation of the training workflow. It seems really interesting, and I am actually quite sure that these training opportunities, they will interest really our funded projects here. Let's hope that this will become a true course that we can arrange with you. And GBIF goodies sounds very good. I do not know what they are, but it sounds good. What I really like here is that you are creating these possibilities in all continents, and also in these countries with they have less their own capacity to produce something like this. So this is really nice. So anybody have any questions for Melissa, please? And if you can see hands, I cannot see hands from here. So please just refer to me. This is really a course that we are now planning to do with together with GBIF, and we have to start thinking about how we can arrange it. But your presentation was very clear, so now we will know the basics how to do it with you. Okay. I do not see any hands here or anything in the chat right now. Let's see. No, I think everything was very clear here.
I think Dimitri has a question.
Okay.
Oh.
It's not really a question. Thank you, Melissa. I just want to add that since 1 year, there is also some former or regional support team in Europe, and also for all the people of the living in a GBIF member country, there is also a GBIF node, and normally they should be very helpful in helping you in publishing data from GBIF. I think we would all like to ask you for help, Melissa, but for practical purposes, we also have a lot of support available in GBIF. And if I look around here, we have Dag already also in this meeting, who is the node manager of GBIF Norway, also very helpful. We have also, in all the GBIF countries with the nodes, there is support, and for the non-members, there is a support team. So also think about asking help a little bit closer to lower the burden on Melissa on helping you all and publish your data.
I just thanks Demi were mentioned about, there is an Europe and Central Asia, we call it ACA support team from GBIF, and here is, I just pasted their email to you, and I thought I heard that they will also organize an office hour, but I'm not sure when. So you can contact them if you have any question, or if you're interested in join their office hour also. And welcome to join both Asia support team and ECA support team. So yeah, just get your email with that.
Okay, yeah, thank you. Anybody else? Any comments or questions or anything to ask from Melissa? This is your chance. I think I saw one hand, or did I didn't see, and you, yeah, and you have given the email address here. So, okay, yeah, thanks, Melissa. I think that we are now reaching very soon the end of this nice workshop. And we are going to have one more Menti, and it will be a quite relaxed Menti because it will be a free-form Menti, where we are just expecting some of your words. Celine, could you share it? And I will once again, I will have to put the Menti link there because in case, because we have so many words already there in the chat box, so that I will have to rewrite it again, just, or I am just too slow. Okay, it's here. What are the key messages that you will remember this from this workshop? And let me explain this a little bit what we mean here. Now you have heard all these great presentations. You have discussed with the presentators. You have discussed with each other here. What kind of things have stuck in your mind from here? And is it the diversity of principles? Is it the diversity of actors? Or is it just like Jennifer said, or is it avoidance of chaos? And on what data management issues you are possibly going to concentrate next? You can write here short sentences. You can write here only words. Whatever comes to your mind, what are you going to do next? What's on your mind after now? And we are going to have this nice looking cloud of words and nice looking cloud of sentences. Thank you very much. I think we can go now basically for the final words of today and the next steps on tomorrow and future. Oh, we had additional Menti as well. Okay, how much? Yeah, well, this is a simple one basically. How much do you think you have to update your DMP in the next couple of months? This is all. These numbers are. They are actually producing quite interesting knowledge and quite interesting data for Biodiversa as well, because we are reorganizing this workshop also for the next call and the call after that. So it's always interesting to see how effective this workshop was. Yes, it seems that most of you think that you have to do some moderate improvements into your DMP, but also quite many of you think that you have to do a lot for your DMP. Yeah, moderate seems to be the winner. So thank you very much. If you have any questions or if any comments, you can email us.
Thank you, Harri. Thank you, Gaby. Thank you, all speakers.